Start.
Good morning, students. Welcome back to Botany second year plant physiology class, chapter 4, photosynthesis in higher plants. Already you have learnt about light reactions and dark reactions, where glucose is prepared utilizing carbon dioxide and water. And in the dark reaction, generally we refer dark reaction, the biosynthetic phase, you have seen Calvin cycle. Melvin Calvin, the scientist, has discovered it. In the previous class, you have learned about the Calvin cycle, which comprises of 3 stages. That is carboxylation, where carbon dioxide is going to combine with RUBP in the presence of Rubisco enzyme. Then reduction, where the first stable compound, phosphoglyceric acid, PGA, the 3-carbon compound, is going to form, and it undergoes reduction. After undergoing reduction, phosphoglyceraldehyde is going to form, and from those phosphoglyceraldehyde molecules, 2 molecules are going to form the starch or the glucose, and the remaining 10 molecules of phosphoglyceraldehyde is going to go for the regeneration of RUBP, which is a 5-carbon compound. So here, let us have the quick part of this appreciation. After completion of the Calvin cycle, many of the children, they forwarded their work, and I was very happy because very soon they are completing the work and forwarding it to me. G. Pranita Abhinayasri from Vangara, TSRJC. J. Sandhyarani Ramya Karishma, S. Akhila Y. Nagasri, R. Triveni from Vaira, TSRJC. Very good children, keep it up. Keep the spirit of learning like that only. So here in the previous class, we completed light reaction and Calvin cycle. So once we have to revise it, so that today's topic is photorespiration. Photorespiration means in the presence of sunlight, the respiration is going to take place. Respiration, as you know, utilization of oxygen and liberation of carbon dioxide. So here oxygen is going, which is opposite of photosynthesis. So here in this photosynthesis, the light reaction and Calvin cycle, C3 cycle, just we'll have a quick review here. As you can see, the light getting harvested by the light harvesting complex, the chlorophyll A molecule trapping the solar energy, light energy, and splitting the water molecule, releasing oxygen. As you can see in photosynthesis, it happens in the light reaction, as well as ATP and NADP H are going to form here. So here ATP is going to form when adenosine diphosphate is going to combine with inorganic phosphate. So ATP formation that takes place in the stroma. This is a chloroplast, and in this you can see this light color is the stroma. So ATP is going to form, and NADP is going to get reduced to NADPH. So here the reducing power NADPH as well as ATP are the end products of light reaction, and these get involved in the carbon fixation. That means the carbon dioxide getting utilized, and it is going to form the sugar here. So here ATP and NADPH is a connecting link between light reaction and dark reaction, or light reaction and Calvin cycle, as you can see. So this is a Calvin cycle discovered by our scientist Melvin Calvin, and here only 1 carbon dioxide you are seeing, but as you know, in the photochemical reaction, you will see that 6 molecules of carbon dioxide combining with 12 molecules of water and forming the single sugar glucose, C6H12O6, single molecule of glucose is going to form here. And this Calvin cycle, once again we will have a recap in the next slide. C3 cycle, because of the product formed is phosphoglyceric acid, PGA, 3-carbon compound is going to form. And what happens here, you will be having 3 stages: carboxylation, reduction, and regeneration. 3 stages will be there in the Calvin cycle. In previous class, I have shown you about 1 carbon and 3 carbon dioxide molecules combining and undertaking this cycle. But here, completely I'll take the 6 molecules of carbon dioxide when they are going to combine with RUBP. RUBP is ribulose bisphosphate. Bisphosphate means biphosphate, 2-phosphate. You can see here P and P. And it is a 5-carbon compound. You can see this purple color, 1, 2, 3, 4, 5. These are the 5 carbons and 2-phosphate. This is one molecule of RUBP. Like this, how many molecules? You can see here 6 molecules of RUBP combining with 6 molecules of carbon dioxide. Okay. So here 6 6 are 6 5 are 30. 30 carbons will be there here. Okay. And here 6 carbons. 36 carbons will be there. So here finally what will happen, carboxylation means when RUBP and carbon dioxide will combine, it happens in the presence of an enzyme here, Rubisco enzyme. Rubisco, that is ribulose, carboxylase, oxygenase. Carboxylase means when carbon dioxide is combining with Rubisco, it is going to act as carboxylation. That means combination, CO2 and RUBP are going to combine and the carboxylation take place, and it results in the formation of 12 molecules of phosphoglycerate. 12 molecules. And here each phosphoglycerate phosphoglyceric acid molecule will have 3 carbons and 1 phosphate. 3 carbons and 1 phosphate. As you can see here, for RUBP 2 phosphates are there for one single. That means for 6, how many it will be? 12 phosphates will be there. 6 to the 12. So here the 12 has to be given to 12 molecules of PGA. So each one is getting only 1 phosphate and 3 carbons. So like this 12 phosphoglycerate or PGA, phosphoglyceric acid, is going to form. Next, the reduction phase where the end products of light reaction ATP and NADPH are added, and here 12 ATPs and 12 NADPH is going to get added. And when ATP is added, here you can see it is going to become ADP and inorganic phosphate is separated. And NADPH, when it is added, you can see it is going to form NADP plus. And here in this manner, when this glycerate is added with ATP and NADPH, it is going to get reduced and it will become glyceraldehyde. So this phase from carboxylation and reduction to reduction is called, here the acid is getting converted to glyceraldehyde. Phosphoglyceric acid getting converted to glyceraldehyde. Glyceraldehyde 3-phosphate. As you know, 3 is nothing but the carbons only. The carbons are same only here. And the phosphate is also same only. 1 phosphate, 3 carbon. So here 12 molecules of glyceraldehyde 3-phosphate is going to form. Of this, yes, of this 12 molecules of glyceraldehyde 3-phosphate, 2 molecules of G3P. G3P means glyceraldehyde 3-phosphate is going to make the sugar, or the glucose and other carbohydrates are going to get synthesized by using the PGAL. Also we can say PGAL means phosphoglyceraldehyde. Don't get confused with the name. Phosphoglyceraldehyde. PGAL also the same only. Glyceraldehyde 3-phosphate only. Remaining 10 molecules of glyceraldehyde, they are going for the next process called regeneration. Again and again they must regenerate RUBP. RUBP which has 2 phosphates. But here you can see it is not possible because it is going to have single phosphate, monophosphate. Here we will get 6 molecules. 6 molecules of ribulose phosphate. How we will get 6 molecules of ribulose 5-phosphate means, ribulose 5-phosphate means, here of this 10 G3P. 10 G3P means 10 into 3 carbon, how many? 30 carbons will be there. Okay, 30 carbons will be there. And those 10 molecules will have 10 phosphates also. But here from this 30 carbon molecules, we must get a product which is called ribulose phosphate, which is having 1, 2, 3, 4, 5 carbons. That means 10 3 to 30 carbons are there here. 30 carbons, if it is forming a molecule having 5 carbon, means how many? 6. 6 ribulose phosphate will form. 6 5 the 6 5 the 30. Okay. So here the possibility from 10 molecules of G3P is that 6 molecules of ribulose phosphate, each one comprising of 5 carbon and single phosphate. But you know that 10 phosphate are there here. 10 molecules of G3P means 10 molecules of phosphate will be there. but only 6 molecules of ribulose phosphate is formed. Remaining 4 phosphate will be released. It will be given off. It is not mentioned here, but it is given off. Those 6 phosphate are given off, and they want to become biphosphate. Bisphosphate means 2 phosphate they want. So they will lose those 4, and what they'll do, they'll be combining with 6 ATP. So 6 ATP will be added here. So that it can become bisphosphate. So you can see here during this carboxylation to reduction phase also ATP is added. From regeneration of phosphoglyceraldehyde to ribulose bisphosphate also 6 ATP is added. Totally 6 plus 12, 18 ATPs are utilized here. Okay, remember this 18 ATPs are utilized for all the 6 Kelvin cycles taking place for each molecule of carbon dioxide. So here overall 6 molecules is displayed here, but like the 6 cycles will be taking place because 6 molecules of carbon dioxide means each carbon dioxide molecule will undertake 1 Kelvin cycle. So final output is sugar molecule formation and regeneration of 6 ribulose bisphosphate is taking place. So this is the main intention of DOC reaction, where the sugar is also formed here, the glucose is formed, as well as RUBP is also regenerated. So this is our oxidation which you are aware of light reaction and dark reaction over. So photosynthesis process is done for you. Now today's topic is photorespiration. For this you must know once again the importance of Rubis coenzyme. Rubis, ribulose sugar, carboxylase, oxygenase enzyme. C for carboxylase, as you have seen the role in the C3 cycle, this Rubisco is going to make the carbon dioxide combine with RUBP, or it may make the RUBP combine with carbon dioxide. That's why the carboxylation process took place in the Calvin cycle. Rubisco. Rubisco is having 2 roles because it can act as carboxylase, it can act as oxygenase. Whenever carbon dioxide is more, immediately it will go for this one. It will make the RUBP combine with carbon dioxide. And the phosphoglyceric acid is going to form and sugar is going to form. Again, regeneration of RUBP is taking place. Okay, this is the photosynthesis that you have learned. But here, what you are saying, photorespiration nature also is there. Photorespiration means how it will take place. In the presence of light only, when the temperature is when the oxygen is more, this oxygen is going to combine with Rubisco. Sorry, this oxygen is going to combine with RUBP in the presence of Rubisco, and it is going to form 2 compounds here. The 2 compounds are, one is phosphoglyceric acid, okay, and another one is phosphoglycolate. This phosphoglycolate is 2 carbon compound. PGA is 3 carbon compound. You can see here 2 PGAs, 3 carbons, 6 are there. Here there is no carbon here in the oxygen. So here only oxygen is coming. So RUBP, which is a 5 carbon, will be dividing into 2 molecules, one having 2 carbon, another one having 3 carbon compound. Okay, for 3 carbon compound, the PGA will be going for the regeneration of RUBP like this. Okay, but what about this PG, phosphoglycolate? This one, which is having only 2 carbon, it will combine with another phosphoglycolate. That means 2 plus 2, 4 carbons will be there. So in that manner, it can lose 1 carbon dioxide. And here it is utilizing ATP and NADPH, and 1 carbon is removed here. So again, 1 more PGA can form here, and it will go for the RUBP regeneration. So this thing is nothing but photorespiration. Because of lack of 1 carbon molecule, because oxygen is combining, carbon dioxide is not combining, oxygen is combining with RUBP, we are getting here phosphoglycolate, which combines with another phosphoglycolate and loses 1 of the carbon in the form of carbon dioxide. It can be considered as photorespiration. It occurs in the presence of sunlight. That's why it is photorespiration. And this phosphoglycolate combines with another phosphoglycolate and loses the carbon in the form of carbon dioxide, and again it will get transformed into phosphoglyceric acid. So here whatever the reactions are taking place here in this point is considered as C2 cycle. It is considered as C2 cycle because phosphoglycolate is 2 carbon. This 2 carbon and another 2 carbon, like that only phosphoglycolate, are going to combine and become 4 carbon, and it will be easy for them to lose that 1 carbon. Okay, and they are going to form again the PGA, which is having 3 carbon. Join back the RUBP regeneration process. Okay, so this is the thing happening in the photorespiration. So remember that this is a wasteful process because simply it is utilizing here ATP and NADPH. It is utilizing ATP and NADPH, as well as it is liberating carbon dioxide. This carbon also is going to get utilized in the photosynthesis back again. So 2PG, 2 carbon compound phosphoglycerate is going to become phosphoglyceric acid. This is only the photorespiration phase where C2 cycle is going to take place, that you are going to learn. I hope you are understanding this nature of Rubisco, the most abundant enzyme in the world. Its active site can bind both carbon dioxide and oxygen. Rubisco has greater affinity for carbon dioxide than oxygen. Thank God it is there, because it is this greater affinity of Rubisco to carbon dioxide synthesizing the glucose molecule. Generally, Rubisco acts as carboxylase, but when the temperature increases and the concentration of oxygen increase, it acts as oxygenase. So, due to high temperature and increase in concentration of oxygen, the Rubisco is going to act as oxygenase. This is very important point. So, NEET point of view also, some of the important questions will be asked from this topic only. So, listen carefully. So one more time, you can see the nature of Rubisco enzyme here, well described in this picture. RUBP combining with carbon dioxide in presence of Rubisco, carboxylase, forming 2 molecules of glyceraldehyde phosphate or phosphoglyceric acid. From these 2 will be utilized for sugar production or glucose, and the 10, which is going to convert it to phosphoglyceraldehyde, which we call triose phosphate also, is going to form the regeneration of this RUBP. Okay, this is a good process. That's why here a smiley is there. And you can see in this process, we are using ATP and NADPH. And here for 6 molecules of carbon dioxide, 12 ATP plus here 6 ATP, 18 ATPs and 12 NADPH will be utilized. Okay, and here we are going to synthesize the sugar here. Sugar is going to form. You can see the light reaction ATP and NADPH end products are utilized here. That means the chemical energy from the sunlight will be fixed into this sugar, glucose, which we are going to have the chemical bond energy here. The radiant energy is converted to chemical bond energy, that is the sugar. Now coming back to this Rubisco, the oxygenase nature. When the temperature is high and when oxygen is more, at the time, this RUBP is going to combine with oxygen in the presence of Rubisco, giving 1 phosphoglyceric acid, 1 PGA, having 3 carbon, and 1 phosphoglycolate. And utilizing ATP and NADPH, losing 1 carbon dioxide molecule, again it will become phosphoglyceric acid only and goes for the RUBP. Okay, so here this part is called the C2 cycle. At this point, the C2 cycle will be taking place. C2 cycle is taking place with this 2 carbon compound phosphoglycolate. Now let us learn about this phosphoglycolate, how it is going to undertake that C2 cycle. Sorry. So here, definition of photorespiration, also known as oxidative photosynthetic carbon cycle. Carbon cycle only is taking place here, and it is oxidative because of availability of oxygen. And in the plant part, it is taking place. That's why it is photosynthetic carbon cycle. because further the PGA formed will be going for the regeneration of RUBP. That's why we have to mention it is photosynthetic carbon cycle, oxidative photosynthetic carbon cycle, or simply we can say the C2 photosynthesis. It is a process in plant metabolism which attempts to reduce the consequence of a wasteful oxygenation reaction by the enzyme Rubisco. So the Rubisco enzyme is held responsible for the C2 cycle. Okay, and the consequence can be reduced by carrying the C2 cycle. This C2 cycle is going to get back again into phosphoglyceric acid and go for the photosynthesis, or the process that leads to only one 3-PGA being produced or produced in the dark reaction in chloroplast. So here simply we can say only one 3-PGA or glyceraldehyde 3-phosphate phosphoglyceric acid is going to form, only one, and the another one is 2-carbon compound phosphoglycolate. So this is only photo respiration. Now let us see in this picture and understand the process how photo respiration takes place. So here this dot method, you can see carbon, these two blue color, it is nothing but the carbons. You can see 2 carbons are there here. First, what is happening? First, RUBP, which is having 5 carbons, due to the oxygenation nature of Rubisco, the oxygen is going to combine with RUBP, forming 1, 2. 2 compounds are formed here, 1 and 2. The first one is phosphoglyceric acid, having 3 carbon, which will be going for the reduction phase and form phosphoglyceraldehyde and regeneration of RUBP. This one is C3 cycle only. Here you can see this is the C3 cycle. But what about this another molecule, phosphoglycolic acid, having 2 carbon? It will be combined with another phosphoglycolic acid. 2 plus 2, how many? 4 carbons will be there. From that 4, it will be easy for that to lose 1 carbon in the form of carbon dioxide. That means again it will be having 3 carbons. 3 carbon compound is nothing but phosphoglyceric acid. So this is only our C2 cycle. I hope it is clear. Look at the diagram properly and try to understand. RUBP, oxygen, Rubisco enzyme, and 2 products. Phosphoglyceric acid 3 carbon compound, phosphoglycolic acid 2 carbon compound. Phosphoglyceric acid 3 carbon compound because of that carboxylation, because it is combined with carbon and forming 3 carbon. So here we will see PGAL aldehyde is going to form by reduction and regeneration of RUBP taking place. Okay, this is C3 cycle, common thing that we can see. But here the phosphoglycolic acid is going to take C2 cycle. That means another phosphoglycolic acid which is having 2 carbon, both of them will combine 2C plus 2C, 4C. From 4C it is easy to lose 1 carbon in the form of carbon dioxide. And again the 3C compound is nothing but phosphoglyceric is formed here. Now C3 plants. C3 plants means the plants which will undertake Calvin cycle, they are called as C3 plants because the first form stable product is phosphoglyceric acid. In C3 plants, some oxygen does bind to Rubisco and hence carbon dioxide fixation is decreased. So the drawback in C3 plants we are talking, the drawback is generally it will undertake Calvin cycle. But sometimes the oxygen binding capacity of Rubisco will make it take photorespiration, which result in decreased carbon fixation, decreased glucose formation. The formation of glucose or photosynthesis rate will be somewhat decreased here because of this nature. RUBP, instead of forming 2 molecules of phosphoglyceric acid, binds with oxygen and forms 1 molecule of phosphoglycerate and 1 molecule of phosphoglycolate in a pathway called photorespiration. So Rubisco will be making RUBP combined with oxygen, not with carbon dioxide. It will make RUBP combined with oxygen, forming 2 molecules. One is phosphoglyceric acid, that is phosphoglycerate, and the another one is phosphoglycolate. in a pathway called photorespiration. So in this photorespiratory pathway, there is neither synthesis of sugar nor ATP formation is taking place. That's why it is a wasteful process. There is release of carbon dioxide. That's why it is called respiration, utilizing ATP. It is a wasteful process. So this is the most important picture where you are going to see how the C2 cycle or photorespiration cycle is taking place. As you are aware of this chloroplast, it is present in the plant cell. There are peroxisomes present in the plant cell and mitochondria. All these three are important for undertaking photosynthesis. As you can see here, the granum thylacite green color is there, and this is the stroma whatsoever. Here we will be seeing the C3 cycle taking place. But C3 cycle will be taking place generally. But what is happening here? Temperature has increased. Oxygen has increased. Concentration of oxygen is increased. Now, the enzyme Rubisco is going to become oxygenase in nature. So it will be making the ribulose 1,5-bisphosphate RUBP. Ribulose 1,5-bisphosphate means RUBP. Means 5 carbons, and to the first and to the fifth carbon, the phosphate is attached. 2 phosphate will be there. This RUBP and oxygen will combine, and they are going to form two products. One is the phosphoglyceric acid, another phosphoglycolate. And the most important thing, notice here, I am taking here 2 molecules of oxygen and 2 molecules of RUBP. Why it is mentioned here 2 molecules of oxygen and 2 molecules of RUBP? Oxygen will form 3 carbon compound, phosphoglycolate 2 carbon compound. And further, one more phosphoglycolate must be available. Then only the further C2 cycle will take place. This is the thing for taking these 2 molecules of oxygen, 2 molecules of RUBP. RUBP having 5 carbon. 5 into 2 how many? 10 carbons. And here the same RUBP and oxygen combining and forming 3 carbon. Another 3 PGA is having it is having 3 carbon. Okay, so 2 molecules of PGA is formed, and here 2 molecules of phosphoglycolate is formed, each one having only 2 carbon. As you can see clearly, it is mentioned 2 carbons, means 2 carbon and 2 carbon. You can see both are separated only, but they are there in the chloroplast. I hope you are understanding the number of molecule count and carbon count clearly without any confusion. So what will happen here? PGA is going to undertake the Calvin cycle because it is PGA, 3 carbon compound, 2 molecules are there. So they'll go for the sugar formation, regeneration, reduction, regeneration, next steps of Calvin cycle. But this phosphoglycolate, it is having 2 carbons. First of all, it will try to lose the phosphate. Both of them, both the 2 carbon and 2 carbon, they are going to try to lose the phosphate. Okay, so here you can see the phosphate is released here. When the phosphate is released, the glycolate is only present now. Now we are having 2 molecules of glycolate. Now both these 2 molecules of glycolate is going to enter peroxisome. In this peroxisome, this glycolate, you can see it is getting reduced. Here, when oxygen is available, it is oxidized. First of all, it will get oxidized. When oxygen is available from the glycolate, the water component and the oxygen will combine and hydrogen peroxide will form. Hydrogen peroxide is going to form, and this glycolate is becoming glyoxylate. So clearly see here, both the glycolate molecules coming from chloroplast to peroxisome, immediately with the availability of oxygen, they are going to become glyoxylate. They are oxygenized. That's why the name glyoxylate. And also hydrogen peroxide is also formed here because of oxygen available. So that we leave it here only. We are bothered about this one only. Now further this glyoxylate, it is going to become glycine. It will become glycine. How it will become glycine? Here that diagram is not there. Amine group. One amine group will be present here. Amine group is going to get added to this glyoxylate. At that time it will become glycine. After getting added with the amine group, the glycine, immediately it will move to the next part. That is the mitochondria. In the mitochondria, this glycine, two molecules, which are having 2 plus 2, 2 carbons are there for each glycine, no? 2 plus 2, 4 carbons will be there. From these 4 carbons, one carbon dioxide, one carbon is removed in the form of carbon dioxide, as well as it is going to make the NAD get reduced to NADH2. Okay, NAD getting reduced to NADH2. Remember this also. And you know the glycine is also having the amine group. The amine group will be as it is. Here it is shown as if it is released, but if it is released also, it is going to enter into the peroxisome. You can see this glycine is now become serine. This serine of it is going to leave the mitochondria and enter back into the peroxisome. When it will enter into the peroxisome, at that time only this amine group, which is already present, that will be released here in this peroxisome. So here a thing which you have to remember is one amine group is going to combine with the glyoxylate, and it is becoming glycine. That glycine will enter here and it will be getting it will be losing one carbon in the form of carbon dioxide, as well as it is getting converted to serine. Meanwhile NAD is getting reduced to NADH2, and this serine is going to enter into the peroxisome. Here this serine is going to lose this amine group. So amine group, you have to remember the amine group, this amine group NH3 amine group is going to combine with glyoxylate, make the glycine formation, again enter into the mitochondria, again through the serine it is going to get released into the peroxisome back again. Okay, so this amine group is going to move between peroxisome and mitochondria, remember that thing. And this serine is this 3 carbon compound. Now because it is losing one carbon, the two molecules of glycines, 2 plus 2, 4 carbons were there. Of that 4 carbons, only 3 carbons is left over. So this serine is having a 3 carbon compound. It is entering the peroxisome, where you can see it is going to become hydroxypyruvate. Why it will become hydroxypyruvate? Because it is losing the amine group here. It is going to become hydroxypyruvate. Now when it is becoming hydroxypyruvate, at this moment you can see NADH2 will be added and it is going to become NAD, and it will make it form the glycerate. That means the ions are removed here and glycerate formation is taking place. This glycerate will enter back again into the chloroplast, where while entering in the glycerate enters into the chloroplast, immediately ATP will be added. ATP means phosphate is added. When phosphate is added, it will become phosphoglycerate or phosphoglyceric acid. That is nothing but PGA. You can see in the beginning the 2 carbon compound phosphoglycolate is losing the phosphate and going to the peroxisome, mitochondria, and again getting transformed and again coming back to the chloroplast. Again it is added with ATP. That means it is phosphorylated, glycerate is phosphorylated and phosphoglyceric acid is formed here. So this is nothing but our C2 cycle. I hope you understood this one. Once again, I will explain the same thing. Listen, two molecules of ribulose 1,5 bisphosphate. Each one having 5 carbon means 10 carbons will be there here. And two molecules of oxygen combining, forming 2 molecules of phosphoglyceric acid. Each one having 3 carbon, 3 plus 3, 6 carbons over here. And one more compound, it is forming 2 molecules of phosphoglycolate. Each one having 2 carbons. 2 carbons plus 2 carbons for 2 molecules means 4 carbons are there here in the chloroplast. It is losing the phosphate. 2 phosphate it is losing. Only carbon molecules are there. It is now called as glycolate. It will enter the peroxisome. In the peroxisome, on oxygenation, it is going to become glyoxylate. And it also forms hydrogen peroxide. And this glyoxylate having 4 carbons now becomes glycine. How it becomes glyoxylate to glycine means amine group is added here. NH3, ammonia is added. And that amine group is going to make that glycis now enter into the mitochondria. When it enters the mitochondria, immediately the 4 carbon compounds will be there, no? From that, 1 carbon is lost in the form of carbon dioxide. And it also makes the NAD become NADH2, get reduced, as well as serine is going to form. Now this serine, which is having the amine group, enter back into the peroxisome. This serine, after entering the peroxisome, loses amine group, and now it is becoming hydroxypyruvate. This hydroxypyruvate, by the reducing power of this NADH2, becomes glycerate. This glycerate further moves back to the chloroplast, and this glycerate on phosphorylation by addition of ATP becomes glyceric acid, phosphoglyceric acid. So these 3 carbon compounds, serine, hydroxypyruvate, glycerate, phosphoglyceric acid, this is a 3 carbon compound is formed back again here. So what the thing you are seeing is carbon dioxide is liberated here in the mitochondria. This carbon dioxide will be further used in the photosynthesis, no problem. But here, one carbon dioxide is removed here. And because ribulose bisphosphate is combining with oxygen, the problem has started. The problem was nothing but it has to combine with carbon dioxide for forming of phosphoglyceric acids, 2 molecules. But it is combining with oxygen, so it is going to form 1 PGA, 1 phosphoglycolate. 1 phosphoglycolate will have 2 carbon compound. So this 2 carbon compound phosphoglycolate will combine with another phosphoglycolate. So here 2 phosphoglycolates are going to combine, and slowly they are going to move to the peroxisome, mitochondria, losing the carbon dioxide and getting back to the peroxisome, coming back to chloroplast and becoming the phosphoglyceric acid, which is a 3 carbon compound. So this cycle is called C2 cycle, or this is also considered as photorespiration, where oxygen will be utilized, carbon dioxide is liberated. Okay, so this is a thing very important, neat point of view also. Once again, you can have clear cut vision of the C3 cycle and C2 cycle. The nature of RUBP. RUBP, if it is combining with carbon dioxide, it will become phosphoglyceric acid. 3 PGM is 3 carbon compounds. Okay, it is not a molecule. It is only 3 carbon. 3 number is not the molecule. It is the 3 carbon here indicating phosphoglyceric acid. It is going to undertake the reduction. When it become reduced, it will become triose phosphate. Phosphoglyceraldehyde is nothing but the triose phosphate. From this triose phosphate, 2 molecules will be utilized for the sugar formation. Remaining 10 will be utilized for the regeneration of RUBP. Whereas in C2 cycle, this RUBP is combining with oxygen. And so when it combines with oxygen, 1 PGA will form. That will go for the regeneration process only here. But another compound is phosphoglycolate, 2 carbon compound. Now the journey of phosphoglycolate will begin from chloroplast, and it will become glycolate, glyoxylate, glycine, serine, pyruvate, glycerate, and convert back to phosphoglyceric acid. So all these things which are taking place here, the changes are taking place from chloroplast to peroxisome to mitochondria. Again from mitochondria to peroxisome, peroxisome to chloroplast. Back again, it is going to become a 3-carbon compound called phosphoglyceric acid. So this is C3 and C2 cycle. I hope you followed this very well. You understood very well. Look at the diagrams, draw the diagrams. If you draw and write the diagrams, you'll remember the names of the compounds forming, as well as it will be easier to understand without any confusion by just looking at the diagrams. Now C4 plants. Tomorrow's topic is C4 plants only. C4. C4 is nothing but in C4 plants, photorespiration does not occur here. You know that in C3 plants, we are having phosphoglyceric acid, the stable compound forming. That's why the name C3 plants. In C4 plants, they are adapted. They are having a special structure. Chloroplast dimorphism takes place here in C4 plants, which is going to skip this photorespiration, which is a wasteful process. Photorespiration, C4 plants are going to skip. They developed a mechanism to increase the concentration of carbon dioxide at the enzyme site. You know Rubisco, it is having high affinity to carbon dioxide. But when temperature increases, when oxygen increases, it will become oxygenase and undertakes photorespiration. To skip that photorespiration, these C4 plants, they developed a mechanism for increasing the concentration of this carbon dioxide, where at the enzyme site only they don't want oxygen to combine. They want only carbon dioxide to combine with the RUBP. So at the enzyme site of the Rubisco, this carbon dioxide concentration is going to increase and increase. And this takes place when the C4 acid from the mesophyll is broken down in the bundle sheath. So remember, in C4 plants, we will be having the mesophyll tissue as well as bundle sheath are special cells surrounding the vascular bundle. Okay, where Calvin cycle will be taking place, C3 cycle will take place. So here to release carbon dioxide, this results in increasing the intracellular concentration of carbon dioxide. So bundle sheath arrangement is there that you are going to learn tomorrow. In turn, this ensures that the Rubisco functions as carboxylase, minimizing the oxygenase activity. So C4 plants are going to give higher yield, higher productivity because they are skipping this photo respiration. They are having a good mechanism where they are going to form C4 acid, and they are having mesophyll cells, bundle sheath cells. Now assignment 7, describe in brief photo respiration. If you understand this topic very well, the next chapter also will be easier for you to understand. Thank you. Thanks one and all. Sir, class is over. Sir? Sir, class is over.